Welcome everyone to the third webinar of the International Eosinophil Society. We will be hearing about regulatory eosinophils and single-cell RNA sequencing today, both of which are evolving rapidly. Before presenting today's moderator and giving you some practical information about the webinar, I would like to briefly present our society and its missions, especially for those of you who are attending for the first time. The International Eosinophil Society is a scientific society whose goal is to foster collaboration and to promote educational events that are related to eosinophils and eosinophilic disorders. We organize a symposium every 2 years, and due to the current pandemic, we have replaced this year's meeting by a series of webinars that are free to all those who are interested. I encourage you to visit our website to see what we do, and eventually to consider joining us as members. Now for the practicalities of today's meeting. The presentations will last 20 minutes each with no interruptions. And directly after each presentation, we will have 5 minutes for discussion. The speakers will answer questions that you've entered in the question and answer section on the right-hand side of your screens. And you can do this during the presentations. It will be the moderator's task to select the questions and interface with the speakers on your behalf. The presentations will be recorded and uploaded to our website within days after the meeting, and the link for the website will be posted at the end of the meeting today. And now I would like to introduce our moderator, Professor Bart Lambrecht, who has kindly agreed to animate this session. Both subjects that are going to be presented today, regulatory eosinophils and single-cell RNA sequencing, I think, are of interest to his current work. Professor Lambrecht is Professor of Pulmonary Medicine at the University of Ghent here in Belgium, and he is Scientific Director of the VIB Inflammation Research Center. He came to present his fascinating work on Charcot-Leyden crystals at the last IES symposium that was held in Portland 2 years ago, and he honors our society today by being with us for a very interesting session. Thank you very much, Bart.
Thank you very much, Florence. So can everybody hear me? Florence? Okay, so it's my pleasure to introduce the first speaker. It's Professor Aline Bozec. Aline is the chair of internal medicine at the University of Erlangen in Germany, and she will talk about comparative studies of murine eosinophils in the lungs and in the synovium using single-cell RNA sequencing. And this technique of single-cell RNA sequencing, last time when we had the International Eosinophil Society, the general consensus was still, that it was virtually impossible to do. But you will hear during the day that there are now several possibilities, several labs have succeeded in this. And you will also see from the broadness of the speakers, this is coming from departments of internal medicine, anesthesiology, allergy. So this is actually very exciting that eosinophils are on the research agenda across many, many different fields. So it's really my pleasure to introduce Aline. It's going to be a pre-recorded talk, but Professor Bozec is with us today and she'll be able to answer the Q&A live. So please go ahead and play the session, please.
So, good afternoon, ladies and gentlemen. First of all, let me thank the organizer to give me the opportunity to present my work here. My name is Aline Bozec, and I'm working in the Department of Medicine in Clinic 3 in Erlangen, which is specialized in rheumatology and immunology. Today, I would like to present you a study demonstrating that a different subset of eosinophils exist in the lung and in the synovium, and that they have strong action in rheumatoid arthritis resolution of inflammation. Rheumatoid arthritis, as you might know, is affecting the joint of the patient. It's leading to joint deformity and disability. Basically, you have an hyperplasia of the synovium, which is due to the immune cells infiltration. When we talk about immune cells infiltration, we talk about the infiltration of neutrophil in the first set, which is followed by a macrophage and a big chronicity of the inflammation within the joint. This inflammation is leading then to cartilage destruction and bone erosion. This is the latter, the bone erosion and the activation of the osteoclast, which is leading to joint deformity and disability for the patient. Rheumatoid arthritis is an autoimmune disease which is affecting mostly women over 50 years old. And this chronicity, in fact, is a main problem in rheumatoid arthritis, because in the clinic we can dampen the inflammation, but we cannot fully resolve the inflammation. So one of the main goals in my lab is to understand what is inducing this chronicity and how we can force resolution of inflammation. So basically what we have done before is that, in fact, in 2016, we were able to show that by pushing the Th2 response through an infection model, with a warm infection model with Nipostrongylus brasiliensis, which is well known to induce Th2 cells and as well hypereosinophilia in the mice, we combine this infection model with serum-induced arthritis, and we could nicely show that, in fact, we have a strong resolution of inflammation when we have infection with Nipostrongylus brasiliensis. To make a long story short, we could nicely show that, in fact, eosinophils in these models were essential for driving the resolution of Nipostrongylus brasiliensis, induced resolution of arthritis. And in these pictures, I would like to show you that, in fact, when we push the Th2 response, we can as well push the eosinophils to migrate into the joint. And we could nicely see that these eosinophils are infiltrating the joint of the mice over the course of the infection of Nipostrongylus brasiliensis. Of course, this leads to the next question. Since we have hypereosinophilia during asthmatic models, what is the relationship between asthma and rheumatoid arthritis disease? There are several studies showing indeed that there is an inverse correlation between the number of asthmatic patients and the number of rheumatoid arthritis patients. They are likely protected, but how is it working? So in 2016, Martinez al in the GCI paper nicely show as well that they have described two sets of subset of eosinophils. The first one that it names the regulatory eosinophils that present an intermediate level of Syglec-F expression and that is show less activation marker or gradual protein release, which is more for me the resident eosinophils, since they have a lot of homeostatic function. In addition, they actually show that under asthmatic response, what is happening is that the resident eosinophils are replaced by the inflammatory eosinophil, which present high levels of Syglec-F, and active markers in that can drive inflammation and damage of the lung tissue. So one of our main questions was to delineate whether first asthma can resolve rheumatoid arthritis, and second, whether there is several eosinophil subset between the joint or between the lungs that could explain the discrepancies that we see in these two diseases. So this is basically our current hypothesis, that allergic response in an asthmatic model, models will, in fact, together in rheumatoid arthritis murine models, what is happening to the eosinophil subset in the lung and in the joint, and how this is driving the resolution of inflammation by indeed pushing the myeloid cells into an anti-inflammatory profile. In a second aspect, we are as well wondering whether this is ILC2 dependent, but this, I will unfortunately not have the time to present you data about this project. So basically, this is a setting of what we have performed. We have, in fact, induced ANOVA, induced asthma models in a mice, in a biopsy mice, by sensitivization 20 days before induction of serum-induced arthritis. And then we reintroduce an asthmatic hint of asthmatic response by reintroduction of OVA by inhalation. And we analyze the states of serum-induced arthritis. First, we could show that, in fact, serum-induced arthritis is not leading to any difference in an asthmatic response. So we have a nice upregulation of lung eosinophil number in the lung in presence of OVA with or without serum-induced arthritis. We have as well a nice upregulation of IgE or IL-5 in the serum. So our models is working even in combination with serum-induced arthritis. However, what we could nicely see is that in the joint of the mice, when we have a hint of asthmatic response, we could force a resolution of inflammation. And this is represented by the arthritis score, but as well by the number of erosion, which is in fact reflected by the trap positive cells within the histology of the joint. To go a little bit more in to see whether eosinophils are indeed in as well migrating in the joint in these models, we perform first a full analysis of eosinophil I showed you already before, but neutrophil and synovial macrophage or myeloid cells analysis. So the neutrophils are nicely going down when we have an asthmatic response together in the serum-induced arthritis when compared to serum-induced arthritis alone. And we can switch, in fact, the myeloid cell into an alternative activated macrophage state with upregulation of this number of cells in the joint, in the synovial fluid from over-treated serum-induced arthritis mice, which is as well reflected by some qPCR markers. In addition, we wanted to visualize the eosinophil, so we did it by FACS. We have a nice upregulation of eosinophil in the synovial of the mice, and we did it as well by light sheet fluorescent microscopy when we use Siglec-F as a markers. And we can nicely see that in induction of the ova and serum-induced arthritis, we have a strong upregulation of the eosinophil in the joint area of the mice. So when we used eosinophil deficient mice, we wanted to be sure that what we see was fully dependent of eosinophil, and indeed it is the case, because if you look at the dot point here, which are reflected by eosinophil deficient mice, they cannot have a resolution of inflammation after asthmatic response. So then we perform, it's where come mainly the subject of the talk, then we perform single cells RNA sequencing. So indeed, we use the mice treated with ovalbumin and as well serum-induced arthritis, and from the lung and the ankle joint, we isolate, we may perform single cells isolation of the cells, and we sorted by FACS, pre-sorted by FACS the eosinophil populations, which means that we sort the cells with CD45 positive, CD11B positive, and Siglec-F positive. And we perform the Illumina 10x genomic single cell RNA sequencing analysis. And to our surprise, we didn't have only one or two subsets of eosinophils, but in fact, we have 8 subsets of eosinophils that we could detect through bioinformatic analysis, as presented in this plot. Quite interestingly, when we compared the eosinophil coming from the ankle and the lung, we can nicely see that they cluster differently, meaning that, for example, the cluster 2 is mainly coming from eosinophil, from ankle eosinophil, whereas the others are coming from the lung. So we decided to concentrate our analysis, for example, to analyze a different gene expression, comparing the ankle joint, the main cluster 2, and the one from the lung, which is basilic cluster 0 here, and the cluster 1 here. And when we compare, we have a strong differential expression of genes. Then we look at some target genes in specifically in this subset, and we look at some pro-inflammatory markers such as MP14 or RNS2A, which are known factors for lung inflammation, and we could nicely see that in fact this marker was nicely upregulated specifically and mainly in the lung eosinophil subset. In addition, when we look at anti-inflammatory markers, which are as well known to play a role in rheumatoid arthritis, such as ALOX15, which are as well known factor for resolution of inflammation, we could nicely see that these markers are allocated mainly in the ankle subset of eosinophils. We pursue a little bit this analysis with by comparing the data that was obtained from the GCA paper. So we took some targets that they mentioned in this paper, where they mentioned that the MIF and ITGX6 are representative of the induced or the inflammatory eosinophil markers. And we could nicely see that in our project, we could see that these two markers pro-inflammatory are as well part of the cluster formed in the lung of eosinophils, which are resident or regulatory eosinophil markers such as ER1 or cells, were indeed only mainly present in the eosinophil coming from the ankles. So basically, we rely quite nicely that the publication from the GCI 2016 was indeed reflected in one of these two populations. We as well analyze the two subsets of eosinophil based on the cyclic F levels in our models, in the lung and in the synovium, and we could nicely see that indeed ovalbumin is inducing a strong inflammatory eosinophil subset in the lung, whereas it can induce as well a strong upregulation of the regulatory eosinophil in the synovium of the mice. Based on this observation, we wanted a little bit more to understand what was the difference between these two subsets. So we isolate the two subsets from the IL-5 transgenic mice and we analyze them. And first, we could nicely see that the induced eosinophil, as published before, and the resident eosinophil coming from the joint as well, have exactly the same observation, that this induced eosinophil present more granuloma, more action of pro-inflammatory, more feature of pro-inflammatory eosinophil with these sombrero vesicles, whereas the regulatory eosinophils are more mitochondria features. Regarding metabolic analysis, we perform a seahorse analysis with these two subsets, and we could see that, in fact, the resident or regulatory eosinophils have less glycolytic function, whereas the induced eosinophils have less mitochondrial respiratory, mimicking again that the inflammatory eosinophils are much more active, so rely on the glycolytic pathway and will probably release more easily and more granulocyte granule from protein from these cells. So based on this, we wanted really to be sure that the difference of action of these two regulatory or induced eosinophil in our models. So we isolate them again, we sort them from the IL-5 transgenic mice. And what we did is that we make an adoptive transfer, but locally in the pro or in the joint of the mice directly during the serum-induced arthritis. So in the day 0, we induce serum-induced arthritis, and day 3, 5, and 7, we introduce either 0.5 million of, in one end, induced eosinophil, and on the other end, regulatory eosinophil. And basically, it was really already obvious from just looking at the mice that the induced eosinophil have a strong inflammation when compared to the one injected with regulatory eosinophil. And when we quantified, we can nicely see that, in fact, the regulatory eosinophil injection can lead to a decrease of inflammation. It was not significant, but to our surprise, we could see as well that the inflammatory eosinophil, when we inject them in the joint, could indeed increase a little bit the inflammation of the serum-induced arthritis mice, which is as well reflected here through the number of osteoclasts and through the number of bone erosion. So in the next, we wanted to understand what is the driving really the regulatory eosinophil in the synovium. To understand a little bit more about this mechanism, we took advantage about the IL-5 reporter mice, and we can see that the IL-5 production was mainly performed by the ILC2 cells. In addition, when we analyzed the GATA1 knockout mice and we forced ILC2 by hydrodynamic injection of IL-25 and IL-33 mini-cycles, we could nicely see that this injection of the mini-cycle is inducing resolution of inflammation but that this was dependent of the presence of eosinophil, since in the green here, where is eosinophil deficient mice, we do not see the strong resolution of inflammation as seen in the wild-type mice. It was as well nicely reflected by facts that this ILC2 driving cytokine can indeed enhance the synovial eosinophil number, and as well by Siglec F expression through light sheet imaging, as shown here in these pictures. So coming back to the single-cell RNA sequencing analysis, we were wondering whether, in fact, the asthmatic response can as well differentiate the eosinophil within the joint. So we analyzed by single-cell RNA sequencing eosinophils sorted from serum-induced arthritis mice only and to asthmatic and serum-induced arthritis mice together. And when we compare that, in fact, only focusing on the ankle, right? So we have a little bit less of subset. We got only 5 subsets of eosinophil in the ankle. And what we could nicely see, in fact, is that when we compare the two, we do not have any difference in terms of subset. There is no one subset popping up after an asthmatic response. We have, of course, quite a lot of differential genes that are upcoming that we could analyze, and some of them are representative of anti-inflammatory response, for example, ALOX15 again, or this RET-LG, which is as well a marker of alternative activated macrophage, but is as well expressed in eosinophil, which is representative of anti-inflammation, which was upregulated in the ova and the serum-induced arthritis together treated mice. And we have as well down-regulation in these mice of some pro-inflammatory markers such as IL-6 receptor or the CD33, for example. So basically what we can conclude from this is that, in fact, the cells are really different cells when we have the resident from the eosinophil in the joint and in the lung, and they do not respond the same in the lung and in the eosinophil. So one could hypothesize that, in fact, they are affected in the lung, they go to the circulation, and then they go migrate to the joint. And it's basically what we wanted to look here by flow cytometry. So we delineate that, in fact, there is upregulation of resident eosinophil in the blood of the mice, as well as strong upregulation in the bone marrow of the mice. So we made the hypothesis that this is confirming the hypothesis that they can migrate from different organs. And in addition, we look at the proliferation of resident eosinophil in the synovial, and we could see that by Ki67 positive codification on the resident eosinophils, that we have upregulation of the resident eosinophil by proliferation in the synovial of the mice. So then we wanted to look whether this is as well true in human. So we had a human cohort of a patient with either active with a full chronic inflammation, which representative to be a little bit more technical, a repetitive score of over 2.6 in our clinic. And we have as well patient in remission. When we have patient in remission, it means that they are under treatment. And then we looked at different markers. So we didn't take Siglec F as a markers, since it's not the same in human and in mice, but we concentrate our markers on EIR1, which was one of the anti-inflammatory markers from eosinophil. And we have the markers of eosinophil, the general markers of eosinophil, which is EPX. So basically, when we look at the pictures, we could nicely see that we have much more eosinophil in the remission patient when compared to the active patient, which are more reflected by a lot of macrophage, which is reflected by the CD68 fluorescent markers here. Whereas in the remission synovial patient, we could see nice EPX staining, and if you look carefully, we can nicely see as well that we have we have as well ER1 and positivity, which is fully absent in the chronic and inflammatory patient. So we looked as well and we quantified that, and we can nicely see that the EPX positive, ER1 positive cells are upregulated in the remission phase. What we did as well is that there were several patients, indeed not so many, but 6 of them, or 8 of them now, that we have already included in it, and that have suffering from asthma, and they got a crisis of asthma. And during the crisis of asthma, one of their doctors told them to take mepolizumab. Mepolizumab is an anti-IL-5 antibodies, which is currently in use for treating asthma. But what they didn't know is that this patient was as well in remission for their rheumatoid arthritis. As you can see here, they were in remission over 20 months. And as soon as they took the first mepolizumab, we could observe in the clinic that they have a strong upregulation of the arthritis. So the arthritis flare was coming back when they took the anti-IL-5 antibodies, which is again showing that there is a strong correlation between the level of eosinophil in the patient and as well rheumatoid arthritis remission. So what I did show you today is that in fact asthma can initiate a systemic IL-5 distribution by lung-resident ILC2, and that this IL-5 resident ILC2 will drive upregulation of the inflammatory eosinophil in the lung, with a strong expansion of the pro-resolving resident eosinophil in the synovium, and that this resident eosinophil produce resolving, such as ALOX15 or ALOX5, for example, that will induce the switching of the classical macrophage into an alternative activated macrophage. Quite a lot of things remain to be done. For example, we are currently performing the single-cell RNA sequencing of eosinophil subset in RA patient, taking into account to sort the ERR1 positive cells. And we want to investigate how we can favor the resident eosinophil amplification in the joint without, of course, enhancing inflammatory eosinophil in the lung. We want to analyze in more detail the composition of the protein and the lipids secreted by the resident eosinophil by metabolome analysis. And we are performing preclinical model using the resolving molecules for a therapeutic approach, again, relapse of arthritis. So with that, we will be able to understand that this is a tissue-specific function priming of eosinophil, which is quite important and still remain to be defined. With that, I would like to thank my group, and particularly Daria Andrew, who performed this nice work, all our collaborators, and of course, you for your attention. I would be happy to answer any question.
Okay, thank you very much, Dr. Bozec. So there are a few questions on the Q&A session. The first question is from Timothée Fretlet. So the question is that for the metabolic profiles and immunological functions, to look at these various eosinophil subsets, you have used IL-5 transgenic animals. And the question is, have you ever looked whether they have some resistance against RA? And the second part of that question is whether the IL-5 transgenics demonstrates similar upregulation of regulatory EOs in synovium and lungs as you show with your other mouse models.
Okay, so thanks a lot for the question. So first we use IL-5 transgenic and we perform serum-induced arthritis, and yes, indeed, they are protected against serum-induced arthritis. So with mice, we did it. If I'm correct, the second question was about...
So it's to see whether they have similar induction of regulatory EOs in the synovium as you see with your other models.
So in the synoviums, they have already a really high upregulation of eosinophil and resident eosinophil in steady state, in fact. So they do not respond so much because they are already really high.
Okay, great. So I also have a question for you as I'm scanning a little bit through the others. So the group in Erlangen with Georg Schett, they have published before that interleukin-9 is also a pro-resolution cytokine for some of the arthritis. I believe it was also in a uric acid-induced arthritis model. So do you think the IL-9 there is related to eosinophil biology via ILC2 accumulation, or?
So we looked a little bit more in this direction. That's why we looked at the crosstalk between the ILC2 and the eosinophil, but so far we could not see a clear link with IL-9. We see a clear link with ILC2, but not with IL-9 specifically.
Right, thank you. So there is also a question from Mario Manresa. So thanks for a beautiful talk. Considering the important role of fibroblasts in tissue remodeling in RA, Mario was wondering whether you have considered or investigated what the role of regulatory EOS would be in the maintenance of fibroblast homeostasis.
So thanks for the nice question. This is something that we are doing in collaboration with one of my colleagues, Dr. Hoffman, which is more experienced in the fibroblast, and we are now performing a co-culture experiment. And so far, we have only really preliminary data saying that indeed it might influence a little bit the fibroblast, but it's really not a major phenotype. Okay,
thank you. So Amy Cleon also has a question. So mepolizumab only depletes about 50% of tissue eosinophils. And GSK, has promoted that it only decreases inflammatory EOS. So how do you see those data in this context?
We unfortunately are still investigating the number of inflammatory, or at least the number of anti-inflammatory eosinophil in the synovium of the patient. Unfortunately, we do not have the synovium of the patient treated with mepolizumab, but in the blood, you are right, it's only 50%, but I cannot tell you anything about because we do not have the answer yet for this question.
Right. I mean, certainly in the clinic, if I may comment on that, benralizumab is quite often used also, and it's giving a complete depletion of I mean, it's certainly not something that emerged in the large clinical trials that there would be flare-ups of arthritis. So do you think it's very common, this flare-up on anti-IL-5 therapy or receptor therapy?
Well, in fact, we had a lot of difficulties to recruit the patient because, as I mentioned, there is really few patients having both disease in the same times. You have either asthma, either rheumatoid arthritis. And we are now trying to recruit more of the patient than this age that I present now. But this was quite of an obvious observation and quite rapid response. And it was as well from the selection of the patient that we are trying to see whether as well asthmatic patient, non-eosinophilic, as well different flare of rheumatoid arthritis, like the one related to neutrophil, for example.
Right. So there are a lot of questions to your talk. So I'll pick one from Steve Ackerman. So I think the question that many of us have, how did you get around the technical problem of being able to do single cell RNA sequencing? Any special tricks or tips?
So we sort the Siglec F, low and high. We did it only in mice, which is for the moment easier than in human. We are trying to establish it in human with Siglec 8, and it's a little bit more challenging than in mice. And to obtain enough cells, we have pulled a lot of mice. So it took us quite a lot of effort, I would say.
Right. And I have the final question maybe from Vaclav. Sorry for the people we couldn't do, but did you ascertain the effect of corticosteroids on eosinophil subtypes. So some might be resistant, some might be sensitive, and some of your patients might have been on steroids.
Yeah, you are fully right, and we are now in collaboration with Jan Tuckerman from Ulm, which has a lot of model on glucocorticoid and in murine mice. Basically, we are investigating as well whether the subset of eosinophil are challenged in this setting.
Okay, thank you very much, Aline, for the excellent presentation. And you should read the questions. There's some very good questions in there. Okay, thank you very much.
So please just mention it. If you have questions, just send me an email and I would be happy to answer them. So just contact me if you have more questions. Thanks a lot to all of you.
Okay, thank you very much. So the next speaker is Cynthia Ju from, she's the Vice Chair of Research at the Department of Anesthesiology at the University of Texas, McGovern Medical School. Cynthia will talk about different subsets of eosinophils in liver ischemia-reperfusion injury. So the floor is yours, Cynthia.
Thank you, Bart. Can you all see my slides?
Not the slides yet, so we still see your camera.
Oh, okay.
Yeah, perfect.
Do you see one screen? One slide or two slides?
We see one screen.
Okay. Okay, great. Thank you, Bart, for the introduction. It's my pleasure and honor really to speak at the seminar series here. I'd like to thank Mark, Bruce, and Florence for the kind invitation. My lab has been studying the mechanisms of acute and chronic liver injuries. We stumbled upon this wonder cell type, eosinophils. So the story I brought today is looking at the protective role of these cells in liver ischemia-reperfusion injury. Okay, so liver ischemia-reperfusion injury occurs in multiple clinical situations. For example, during liver transplantation and liver resection. Besides these liver surgeries, it also occurs in other situations such as hemorrhage shock, trauma, or sometimes even after heart bypass surgery. During liver transplantation, the ischemia-reperfusion injury can cause up to 10% of early graft failures, and it can also lead to incidences of acute and chronic rejection. So currently, the bottleneck for liver transplantation, oh, by the way, liver transplantation is the only viable therapy for end-stage liver diseases, where patients have decompensated cirrhosis or liver cancer. Liver transplantation is the only therapy. And the bottleneck of this therapy is the shortage of donor organs. So in fact, like 15% of the patients die on the wait list for liver transplantation. To circumvent this problem, now more and more often, marginal donor organs are used, but these organs are from aged individuals and from brain dead or circulation dead donors, and also from donors with fatty liver. So these marginal organs are more susceptible to a liver ischemia-reperfusion injury. So right now, there's no really clinical modalities to minimize or reduce the ischemia-reperfusion injury. At the time we started to work on this to work on this model, we knew from the literature in terms of the mechanism of liver injury due to ischemia-reperfusion. So there are two phases. One is during ischemia phase, the hepatocyte, the liver cells are stressed. They are they become susceptible to a second hit. And the second hit occurs during reperfusion when when there's a massive influx of inflammatory leukocytes, macrophages, dendritic cells, and lymphocytes, as well as neutrophils. And a major culprit type of cell that causes liver injury in this model is the neutrophils. They're activated and they release extracellular trap, and they produce reactive oxygen species that can directly cause hepatocyte death that are already stressed and really push them off the cliff. So we wanted to figure out if we can identify new players in this mechanism in order to maybe develop therapeutic strategies. So we started by looking at patient samples. We teamed up with liver transplant surgeons here, and we took 2 biopsies from transplanted patients. One is before, one is after the cold storage before transplantation surgery begins. We call it pre-IR biopsy. And the other one is at the end, after the surgery is complete, before they close up the patient, we took another biopsy, post-IR. So the reperfusion time is about 2 to 3 hours. And we first screened, did immunohistochemical staining, and we only found, you can see obviously there are infiltrating leukocytes in the post-IR, but not in the pre-IR. But we noticed that besides the monocytes and neutrophils, there are eosinophils in the liver, where we could only detect sometimes none or a couple in pre-IR tissue samples. But there is a significant increase of the number of eosinophils post-IR. So here is the looking at each individual recipient before and after surgery. There's the increase of the number of eosinophils. And when we look at the correlation of the eosinophil numbers in the tissue, we found it inversely correlates with serum alanine transaminase levels at 1 day after surgery. So the higher the level, suggesting the higher degree of liver injury. But if there are more eosinophils, there are less ALT levels. So this is what we found in patients. There are significant increases, but we didn't know whether it means anything. So we brought this from the bedside to the bench. We developed a mouse model of liver ischemia-reperfusion injury. So we clamped the blood vessels for 60 minutes, mimicking the time that it takes for the transplantation. And then we released the clamps, reperfused the liver for various time points. And then we first wanted to see if we can also see eosinophil infiltration into the liver in the mice. We did in 2 ways. One is by flow cytometry. We define these cells by high site scatter, Siglec F positive, IL-5 receptor positive, and CCR3 positive. As you can see here, we see similar as in patients, we see a rapid increase in number, and it continues to increase up to 24 hours where we checked. And this is by immunohistochemical staining using anti-MBP antibody that generously provided by Beth Jacobson. Similar to the flow, you can see continuous increase of eosinophils with time. So in both human and mice, we saw the accumulation of eosinophils after IR injury. To understand whether it has any role in this model, we depleted these cells by using anti-Siglec F antibody. This is just to confirm the depletion was sufficient from the bone marrow, the blood, and the liver. So, and we were surprised to see that after eosinophil depletion, the ALT, AST levels in the serum were dramatically increased comparing with IgG-treated control mice. And we also looked at liver injury by evaluating histology Here, the lines outline the necrotic areas in the liver, and this is quantification. And you can see there's a significant increase of hepatocyte necrosis when we depleted eosinophils. So this is the antibody approach. We then used the genetic approach using 2 lines of eosinophil deficient mice. One is the GATA1, comparing with BALP-C wild type. Again, serum transaminase levels are increased in the knockout. Necrotic areas are bigger in the GATA1 knockout mice. We also then used FIL mice, again from Beth. The white bar represent the FIL mice, and the black bars are the wild type. As you can see, the knockout mice have significant increase of ALT, AST, and increased necrotic areas comparing with wild type. So we took a step further in this experiment. We wondered if we give the wild type eosinophils to the knockout FIL mice, could we rescue them? So this is represented by the gray bar. After adopted transfer of eosinophils, we can bring the ALT/AST down, and the area of necrosis is also reduced. So all of these data suggest to us that the eosinophils play a protective role in this case of ischemia-reperfusion injury of the liver. And then we wanted to understand how they do that. We are interested in 2 questions. One is how they're activated, and the other one is how they function to protect the liver. And without too much of clue, we did a microarray analysis. We compared liver non-parenchymal cells, so all the cells except hepatocytes. We compared 2 samples, IgG treated and Siglec F treated. So everything is the same between the 2 samples, except one doesn't have eosinophils. And we saw eosinophil specific genes go down, but one gene that took our attention was ST2. And this is a confirmation by qPCR showing anti-Siglec F treated samples has decreased ST2 mRNA level. And when we did ST2 by flow, we saw that eosinophils are the predominant cell type that express ST2. And here we're showing the eosinophils, majority of the eosinophils also express ST2. As you all know, ST2 is a receptor for IL-33. So we wondered if, and IL-33 is considered as alarmin when the cells, especially tissue cells, epithelial cells, endothelial cells, undergo cell death, they release IL-33. So we wondered if IL-33 is released in our model. So we measured IL-33. These black bars are the wild type at different time points. It peaks at 8 hours. And in the knockout mice, it also goes up a little bit higher than the wild type, and it continues to go up. I think this is probably because the lack of soluble ST2 to neutralize IL-33. So then we wondered if IL-33 through activating ST2 on eosinophils play a role in activating these cells to play a protective role. If that's the case, at least we should see that IL-33 knockout mice and ST2 knockout mice should behave similarly to eosinophil deficient mice, meaning that they should have increased liver injury. So we then conducted these experiments comparing ST2 knockout with C57 black 6 again. Oh, sorry. So you can see based on serum alanine transaminase levels and liver histology, there's a dramatic increase of liver injury in ST2 knockout. The same observation with IL-33 knockout, significant increase of ALT, AST, and increase of necrotic areas. But it's still possible that the IL-33 and ST2 knockout mice have increased injury due to some other reasons, not related to eosinophils. So if it is due to eosinophils, we hypothesize that if we give them wild-type functional eosinophils, we should be able to rescue these mice. So these are the experiments we designed where we were going to give adopted transfer wild-type eosinophils naive or IL-33 stimulated ex vivo eosinophils to the knockout mice. So in the ST2 knockout mice, the black bars are mice treated with PBS. The white bars are naive wild-type eosinophils. The gray bars are the IL-33 stimulated wild-type eosinophils. As you can see, if we give the mice either naive or IL-33 stimulated eosinophils, we can rescue them. And this is histology showing reduction of necrotic areas as well. And so this is what happens in ST2 knockout. But if we give the cells to IL-33 knockout mice, you can see in the white bars, if we give them naive eosinophils, we cannot rescue the mice. The ALT and histology showed similar levels of liver injury. But if we give them ex vivo activated eosinophils by IL-33, we can reduce liver injury, showing by reduction of transaminase levels and liver necrosis. And so this was actually confirming that if you give naive wild-type eosinophils to IL-33 knockout mice, because the mice cannot make IL-33 in vivo, it's not going to work. So this result actually further suggests that IL-33 ST2 pathway in vivo is important in activating eosinophils to mediate the protection. So then our question is, what is downstream pathway? When we went back to our microarray analysis, we found that in the samples that don't have eosinophils, the myeloperoxidase level was higher, suggesting there are more neutrophils. And we confirmed this microarray data by staining the tissues with the MPO. Here you can see in anti-CGLAG-F treated samples, there's significant increase of MPO positive cells in FIL mice and GATA1 knockout mice. Comparing with their respective wild-type mice, we saw a significant increase of MPO positive neutrophils. So these data suggest that the eosinophils somehow suppress neutrophils. The question is how? Here we hypothesized that we knew that eosinophils are the main source of IL-4 and IL-13. So we also confirmed by flow cytometry showing that the eosinophils do express IL-4 and IL-13 in this model. So we then neutralized IL-4 and IL-13 to see if they are protective. When we neutralized IL-4, we did not see any difference in terms of liver injury. But when we neutralized IL-13, we saw a significant increase of ALT, AST, and liver necrotic areas. But also, we saw a significant increase of neutrophils. So even though by flow we showed that eosinophils are the predominant source of the IL-13, but we can't be sure that other cells-derived IL-13 is not playing a role. To confirm eosinophil-derived IL-13 is important, we used the eosinophil-specific IL-4, IL-13 knockout mice, showing in the white bars. Again, this experiments showed that if we just delete IL-13 in eosinophils, the liver injury is significantly increased. So far, the data suggests that IL-33 activating ST2 on eosinophils mediate the protective effect of these cells, and the activated eosinophils produce IL-13 to suppress neutrophils. And the last thing we wanted to do is to see if we can connect the dots. And the obvious hypothesis is IL-33 activation of eosinophils through ST2 produce IL-13, which is not new. But we confirmed this by using two types of eosinophils ex vivo in vitro studies. One is culturing eosinophils from progenitor cells from the bone marrow for 14 days. The other one is using fresh bone marrow-derived eosinophils. As you can see in both cases, looking at protein levels and message levels, we can see that wild-type eosinophils can be stimulated by IL-33 to produce IL-13, but not ST2 knockout eosinophils. So with this data, we came up with this hypothesis that is when there's a tissue injury, in this case liver ischemia-reperfusion injury, the IL-33 is released as alarmin, and it activates eosinophils through ST2, which then causes production of IL-13, and IL-13 suppresses neutrophil activation. And at last, I'd like to thank my team, especially 3 talented postdocs who contributed significantly to this project, Yaochun, Yang, and Meng. And I also appreciate and really like to thank the generosity of Beth Jacobson, who provided us with reagents and mice, and her expertise, as well as other collaborators who contribute to the work. And I'd like to thank NIDDK for funding. Thank you for your attention.
Thank you very much, Cynthia. So this was a beautiful presentation and beautiful publication. So there's a question from Mark Rochman. Did you have a chance to look at the type of IL-33 that is released in this form of necrosis and IR? So is it the full-length processed form, or is it some isoform of IL-33 that could be less bioactive?
Well, that's a great question. We haven't looked. Our ELISA kit detects both, so I don't think we can distinguish that yet. But we're currently working on the source of IL-33, and it seems to be the endothelial cells.
Yeah, I have a related question there, Cynthia. So there are some polymorphisms that control soluble ST2, and that might set the tone, you know, for the general IL-33 bioactivity in vivo in humans. So is there anything known in ischemia-reperfusion injury as to this ST2, soluble ST2 pathway, whether it's differentially in people who have a more severe form of ischemia-reperfusion injury?
That I don't know, but that's a very good question. We can certainly measure, but in a different model of in a model of a chronic liver, or not model, a disease, alcoholic liver disease in patients, we have measured IL-33 and ST2, soluble ST2, and there we found it's quite interesting that only in decompensated patients we see a significant increase of soluble ST2 as well as IL-33.
Right.
So
it's a loss of homeostasis. It's a kind of a loss of homeostasis.
Yes.
Okay, great. So Amy Cleon has a question. So great talk. What are the kinetics of the neutrophil and eosinophil influx post-injury? So the kinetics of the neutrophil and eosinophil influx.
So we haven't compared them head by head in this model. In this case, we saw the eosinophils increase not massively, but significantly as early as 1 hour. And I'm sure if we look hard, we can also see neutrophils at that time. In a peritonitis model, the kinetics is neutrophils come in first, and then eosinophils, and then monocytes. So if that holds true for this model, we would predict neutrophils are in first.
Okay, so there's a question from Giovanna Schiavoni. Do you hypothesize how you know, the IL-33 eosinophil axis could protect against IR. So downstream of the eosinophil, no, and just more than neutrophil inhibition.
More than neutrophil.
Do you think it's all due to the neutrophils, or is there anything more? You know, something like a regulatory eosinophil or something, or?
Ah, okay. So we haven't looked deeper. I mean, this is maybe an interesting question to pursue. That what are the eosinophils we have? Do we have a specific regulatory eosinophils? But in this model, because we are looking at a very short time period after ischemia-reperfusion injury, like as early as 6 hours after reperfusion, the neutrophils play a major role in causing that injury. The macrophages, like polarizing macrophages, happens later. We do have a project looking at later time points, but I think for this particular study, it's mainly neutrophils.
Thanks. There's also a question from Rachel Fitzpatrick. So in the gut, where there's a basal level of, you know, microbiome-driven inflammation, eosinophils are present. Could they also be functioning in a similar way to suppress neutrophil infiltration?
To suppress neutrophil infiltration?
Yeah, so you have a baseline eosinophilia in the gut, you know, part of homeostasis. Do you speculate that that could also suppress the neutrophilia there?
Oh, the neutrophil. Actually, we are very interested in the gut-liver axis in terms of eosinophils. But in this IR model, we don't see so you're saying that the eosinophils that come to the liver may not be
Rachel was drawing this parallel, you know, between the gut, where you have a steady state.
Oh, in the gut ischemia.
Yeah,
oh, okay.
Yeah.
Okay.
Right.
No, we haven't done the gut ischemia, but it's probably, yeah, it probably plays an important role.
Okay, thank you. And we have a final question. So thanks from Vitaly Rusinkovich. Thank you for a great presentation. Could you comment a little bit on the recent data showing the presence of Siglec-F positive neutrophils? So you will be targeting those if you deplete the Siglec-F with a Siglec-F antibody. They play a role in myocardial ischemia-reperfusion injury. So is there anything you've looked at?
Actually, we did not look at whether when we treat mice with anti-Siglec-F antibody, we also depleted or reduced neutrophils. But that's an interesting question. And because we were not comfortable with just the antibody depletions, that's why we used the genetic models.
Okay, thank you very much, Cynthia, for an excellent presentation and very stimulating work. So the final speaker of today's webinar will be Sergej Berdnikov. He's an assistant professor of medicine, associate professor of medicine in Division of Allergy and Immunology in Northwestern University Feinberg School of Medicine. And he will show us how you can use single-cell RNA sequencing of eosinophil population to get insights into their development and tissue diversity. So Sergej, thanks for giving this presentation and looking forward to your talk.
I have a message that I cannot start my video because host has stopped it. So, okay, so now I'm good.
All right, great.
Thank you for the introduction, Bart, and thank you all organizers for opportunity to present at this virtual meeting. I feel very honored every time to present for the society. So thank you all in attendance or if you're checking it out later as a recorded session, thank you as well. So my interests in regard to eosinophil biology are in understanding the cells in their actual environment. So I've been always fascinated by this diversity of eosinophil responses. So this is something we've known for years. We accumulated this incredible knowledge on how many receptors they express and to how many different stimuli in their environment eosinophils can respond. So for example, cytokine stimulations, metabolic change, angiogenesis events, extracellular matrix changes, plasminogen system, mucins, growth factors, and so on and so on. So it shows you how potentially incredibly adaptable this cell is. So my true research interests are to understand how it exists in its environment and is able to adapt in different situations. So by now we really appreciate more and more that there are potentially different subsets of these cells in different tissue states. So we now know that there are resident eosinophil subtypes, not only in lung tissue, but essentially systemically in many different tissues that, if you look hard enough, you'll find them. And in different morphogenetic events or inflammation, you start seeing at least more than one subset coming up that potentially have different functions and look differently and have differential marker expression by flow cytometry. Now, big question for me is whether what we see in terms of subsets or changes observed in eosinophils in those tissues and events, are they true heterogeneity? So are they existing predetermined subsets by their origin, or whether this is potentially simple plasticity? So a cell is trying to adapt to changes in that microenvironment, or even if more than one are present in tissue, just simply they're dealing with different compartmentalization events. So for example, think about lung as an interstitial mucosal compartment and an airway space where you can find eosinophils in different states. So we published this a while ago. It just shows that in the same environment in murine lung, for example, so you see that cyclic F low population just sitting as resident in the lung. And if you induce allergic inflammation, let's say in the classical OVA model, and then you start tracking eosinophils by flow cytometry over time, you just clearly start seeing changes in those populations. So you see expansions of certain subsets and appearance of other ones, like cyclic F high subset, for example. They clearly all look different. So residents, as we heard from many speakers now, that they look like more or less simpler cells, simpler donuts. So, and then during active inflammatory events, we see higher segmentation of the nucleus. And in case of cyclic F high subsets, we have a position of vacuoles, which is fascinating. And now something that really got me more and more interested in this subject is just similar subsets just appear, not even in inflammation, but in normal homeostatic events in the same tissues. So lung, for example, in early postnatal development undergoes significant morphogenetic events, so laying down of epithelium in the airway, branching morphogenesis. And you actually see very similar population subsets of eosinophils, morphologically very similar and phenotypically very similar markers. So question is what all of this is about and what potentially different roles are played by the subsets. So to answer those kind of questions, obviously single cell RNA sequencing would be an ideal approach because it allows you to tease apart all those different populations and show which different signatures and functions they may have. So this technique has been used successfully by many groups for multiple cell types, structural and immune, in all kinds of disease and homeostatic situations. As we all know from the last IES meeting, that's been quite a challenge. Many groups actually trying, to utilize those approaches to study eosinophil heterogeneity, and for some reason, this cell is notoriously hard to work with. So this is a slide where I left you off with during the last meeting. So where we had collaboration with pulmonary critical care division here at Northwestern that was really interested in profiling every single immune cell in the lung. So among those profiles, we tried to find eosinophils, but we had mixed impressions. So first of all, we know that the resident eosinophils are not that numerous, right? It's maybe 1% of all hematopoietic cells in normal mouse steady state lung. But what's interesting, some of odd markers were showing up in other populations. So like this green one, for example, in blue. And those markers that we know are related to eosinophil biology, such as like XBP1, ILS, AKZF3, so PRG2, Al5 receptor, and so on. So at that time, it potentially turned that they could be just eosinophils in less differentiated states residing in those tissues. So my understanding of this is evolving, and that's what I'll be showing you in this talk. But it just shows that there is some complexity to it. Maybe it's not all about one defined mature phenotype. So I really put a hard effort to it since the last meeting. So to be technical and methodical and just try to work it out as a technique, essentially. So, and initially in this talk, I just wanted to present the more technical aspects of it, but now it's really excited about all the biology I'm seeing, so I'll share that as well. So we definitely had some success in doing, mouse eosinophil single cell seq. This is a standard 10x genomics pipeline. So there was a question before, what are the potential tips and tricks that you can use in, modifying this pipeline to work for eosinophils. I can't really say that we added too many tricks. I mean, this is really a standard 10x genomics single cell RNA sequencing pipeline. If any special points I have to emphasize is, one is how fast your prep should be. So in all cases, when we succeeded, our preps were fast, definitely under 1 hour. So from bench to the machine, right? Another trick we used is good use of RNase inhibition, because we know those cells are notorious for like RNase contents, and potential RNase, first of all, is low in eosinophils. Second, gets degraded very easily. So we used actually extra steps of addition of inhibitors, not just in suspensions, but also when loading samples on Chromium chips. So we noticed that this was actually making a difference and improving quality of readouts. And the rest is pretty standard, library preparation. From that step, it's fairly stable, and sequencing and data analysis. So how we decided to start on this is take more or less pure eosinophil populations where we have high confidence that these are actually eosinophils. Because the technique is so expensive, we had to be frugal and combine different types of samples just to get as much information out of it as possible. So instead of sorting, we actually first resorted to bone marrow derived eosinophil culture. So this is standard Helene-Rosenberg's protocol, where you take bone marrow homogenates from normal mice, and first you expand early progenitors with additional stem cell factor and fluid ligand for 4 days. After that, you start adding Al5 cytokine to stimulate eosinophil commitment and this de novo differentiation of eosinophils from expanded progenitors. So every other day, you essentially give them Al5 and you change them flask to flask. So until they become more or less mature phenotypically by day 14. So at that point, they look nearly 100% cyclic F positive. So that's what we typically expect. They have red granules. They, by all means, are normal functional eosinophils. And we all widely use them in all our papers and experimental approaches. So I decided to make it more interesting. So I took these cells, so from day 13. So by flow cytometry, as I said, so they're nearly all cyclic F positive. But what's interesting also, a significant portion of them is double positive for Ly6G and cyclic F. And this is something that Helene-Rosenberg also published. You should check it out. So it's an interesting developmental phenotype. So it's represent more or less precursor eosinophil, so which is still trying to reach that stage. So it's a nice mixed, representation of maturing and mature eosinophils. And then we took day 6, slice in that culture, so where they already seen 2 administrations of Al5, which means they're definitely committing. and on the track to become eosinophils. And they're characterized by actually Ly6G expression here, so that's a blue line, and beginning development of all those more mature phenotypes. So, and these are the results of that single cell from that sample. So it was actually very successful and very interesting. You could easily tell apart which one was later portion of the sample, so from day 13, and which one was from day 6. So identified really significant clusters of mature eosinophils in all respects. So they have cyclic F, as expected, CCR3, interestingly ST2 receptor, CAR4, which is interesting marker of tissue eosinophils. And then the second large population, which has all markers of formation of granules, essentially. So EPX, PRG2, so or MBP, and some other markers associated with eosinophil development. So those two collectively would represent this maturing mature eosinophil pool, which was very exciting. So we definitely nailed them. There was more than one mature eosinophil pool, so I will not go into it, but you can see like gradation of development. Now, in regards to early development, it was actually very interesting. Something I would never expect to see in day 6 culture is how indecisive these cells are. You would think they're all committed to eosinophil lineage development, but we actually see very strong representation of plasmacytoid dendritic cells in these cultures trying to develop in parallel. We also see monocytes, we actually see B cells, which is surprisingly, and we see very strong presence of neutrophils. So all in all, it says that, you know, some halfway that culture truly still retains a lot of myeloid potential. Now, TRIB1 I'm emphasizing here because that's a fascinating molecule. So there is a recent paper published in Blood that shows that TRIB is an essential factor for eosinophil lineage development, actually. And if you knock out TRIB in progenitors, you end up with a very confused cell, which has mixed characteristics of neutrophil and eosinophil. So the paper beautifully lays it out. So if you use TRIB1 as a marker, it just immediately lights up all granulocytes essentially developing with eosinophil potential. So that was an interesting observation. So you can definitely see which ones are absolutely committing. And also it shows how close they are to neutrophil phenotypes. So another important point to take from here is transient expression of classical markers we're used to. So EPX, for example, MBP, would only be seen in the granular formation checkpoint. So once they mature, so they go past that, you don't even see those markers anymore, which is fascinating. So all classical markers we're used to, they're being turned on and off. So don't expect to see them pretty much constitutively expressed at all times in those cultures. And that was very interesting finding. So this is just essentially the same experiment, but just further analysis. It shows you all know it by now, is how you determine different subsets. You just look at very specific marker expression belonging high fidelity to those different subpopulations. And based on that, you can classify them, what they are. So this heat map just shows you more complete signatures of those cells and just shows how related they are in culture. And a lot of eosinophil stories definitely clusters together. Interestingly, neutrophils cluster right in the middle of it, together with pre-Eos and eosinophil lineage committed cells. So eosinophil progenitors are sort of outcasts here. So this is this trace, but this is only because I think they have highly proliferative signature matching those other myeloid subsets. So all in all, we identified actually 10 distinct clusters that we tried to follow in all our other analysis of single cells, sort of as a template, because we have high confidence of what they are, and we try to find them in other tissues. So this, what I'm showing here is single cell analysis of normal murine bone marrow. So it's just simply baseline bone marrow taken from normal mice in vivo. So you probably know it, but there is a huge granulocyte presence in steady state bone marrow, especially neutrophils. So what we did, we took all our markers related to granulocyte development and applied them to bone marrow analysis. And there is a huge representation of granulocytes. Interestingly, we saw neutrophil signatures as expected. All of them have it to some extent. We saw this cluster, which expressed TRIP1, which I discussed, which represents eosinophil committing cells or with potential to commit to eosinophils. And beautiful match to mature eosinophil signature we had in culture, only with complete absence of mature eosinophil markers, such as cyclic F and CCR3 and so on. But in regards to other markers, there was actually a really beautiful match to that cluster. So all in all, it was very interesting to me that there is this mix of neutrophil and eosinophil phenotypes that really falls apart and yet stays together. So it completely drove that interest home for me that there is something about this mixed origin of granulocytes in early stages of development. So we look directly in lungs, actually in bone marrow, trying to tease that apart and understand phenotypic differences. And if you look in bone marrow, you see the small portion of mature eosinophils. So again, it's a very small population, which is cyclic F positive and actually IL-5 receptor negative. They stay in beautifully as eosinophils, and we know they're eosinophils. And then there is IL-5 receptor positive population, which is huge, which also happens to be nearly all Ly6G positive. So those are cyclic negative, Ly6G positive, IL-5 receptor positive. By all means, these cells are actually neutrophils. So talk to any neutrophil biologist or read literature. This is classified as classical standard neutrophil. So when sorted and profiled, so you can see they have very simple kind of donut-like appearance, which implies even if there are neutrophils, they are probably still in submature or band stage morphology, which is very interesting. So, and by single cell, it shows that they have this lots of confused phenotypes. You also can appreciate how extensive those populations are. So in color, we're showing male-female populations of eosinophils, and then talk about neutrophil phenotype. You can see how extensive it is. So in aged mice, approaching actually 50% of all hematopoietic cells in the bone marrow. We also found that this is very sexually dimorphic population. So there are differences which flip actually before and after birth. And in aged mice, they become similar. We also sorted, so Fox sorted those neutrophils, let's call them neutrophils for now, but we're giving them alternative name or additional name, so multipotent myeloid cells, because you'll see what's happening in the next couple of slides. And culture-derived mature eosinophils, so from standard bone marrow eosinophil culture. So left of this blue dotted line, you just can see this receptor repertoire, mediator expression, and functional protein expression from those neutrophils. It's incredible how diverse they are. So if you look at the receptors, you realize they go well outside of neutrophil lineage. So they have, monocyte dendritic cell, developmental markers, receptors, and so on. They express all kinds of chemokines and cytokines, and interestingly secrete or have potential to secrete constituent and so of extracellular matrix, such as fibronectin, for example, thrombospondins, kinase and decorin, and so on, collagens. So potentially they have a role in this homeostatic maintenance of the marrow. And of course, eosinophils induced from bone marrow, as expected, would have more type 2 inflammatory signatures, expected chemokine receptors, and so on. So this is something I showed you at last meeting. It was just my last fascinating finding that if you do sort those cells, the neutrophils, and you give them IL-5, and they have plenty of IL-5 receptor, they constitutively express it, they definitely use it. So, and interestingly, by RNA-seq, that was my last observation last time, they induced eosinophil signatures. So all our classical markers that we talk about by RNA-seq are induced in those cells. So now we went further because our obvious thought was, well, maybe there is just contamination with some stem cells or early progenitors from those cultures, and they just rapidly take over and proliferate and generate that phenotype and RNA-seq data. So now we're way more thorough. So we took and sorted those neutrophils from GFP positive mice, so they were GFP positive, and we transferred them to non-GFP wild type naive bone marrow stroma. So they had their proper bone marrow environment to develop. And then we simply track them by flow cytometry. So you give them IL-5, and you see strong induction of cyclic-F positive phenotype. So you can see by day 5, they become what we consider mature eosinophils, and they have red granular staining, and they look proper. So what's interesting, we also found that IL-5 is actually not necessary to generate this phenotype from those bone marrow neutrophil-like cells or those multipotent cells. So just simply them sitting in stroma induces expression of cyclic-F. So what IL-5 does on top of that, it promotes kind of further maturation of those cells into this classical activated side scatter high phenotype. So that's what IL-5 is responsible for, and stroma can just drive it without IL-5 into eosinophil development. So we have it published in Journal of Leukocyte Biology, and our reviewers asked us actually to probe further the potential of the cells, whether it can become something else. And we treated them by request with MCSF, which promoted induction of monocytic phenotype. So we're able to convert those cells to monocytes macrophages. So you can see the trajectory of induction, 3 days in culture. So we treated them with IL-5, so that resulted in both cyclic-F and Ly6G cyclic-F double positive phenotype. And GCSF, which very interestingly did not just result in pure neutrophils, but strongly induced this mixed Ly6G cyclic-F double positive phenotype. Even more interesting, later on, it was spontaneously changing to monocytes macrophages. So GCSF actually did not just simply maintain neutrophils, but promoted all kinds of phenotypic changes. So at baseline by Western blot, those cells have MPO, so as proper neutrophils should, and no EPX. But as soon as you put them in either GCSF or IL-5, interestingly, they lose their MPO content and upregulate EPX. So we're not the only ones actually showing things like that. So if you look in literature hard enough, it's been published in blood that band stage neutrophils can transdifferentiate to monocytes in different inflammatory conditions. Other papers actually show they can even convert to disease. So it shows that what we call neutrophil is definitely more than just neutrophil. So it's definitely a plastic, myeloid, more precursor stage cell. So to sum it up, classically we think of generation of eosinophils as a strict eosinophil lineage, right? So coming from common myeloid precursor and then granulocyte macrophage precursor to EOP, which we know exists. So it's a committed early stage progenitor of eosinophils. And then further it develops into what we know is now this transitional phenotype, cyclic-F Ly6G, and becomes mature, where it can further be subdivided into different types. So interestingly, in the neutrophil realm, so you realize that actually not much is known about true neutrophil progenitor, just like in eosinophil world. We just don't even know what it truly is. So there are a few papers from single cell that came up, but there is no clear consensus. So then we see these cells that everyone calls neutrophils in the mouse world just based on Ly6G expression. And now we show, and others show, that they're definitely still plastic and can contribute to our lineage eosinophils from outside, which is very interesting. And at the same time to generate other cell types. So, and this is what single cell potential is showing, that's why there is such a close relationship to neutrophil subsets, that potentially eosinophil lineage commitment may occur from those neutrophil type cells, which are being present in bone marrow homogenates. So then we asked question, can we use the same markers essentially and what we learned from the bone marrow and apply it to the lung and see if it can help us to actually get a glimpse into the granulocyte tissue biology. So now single cell shows actually how deep the rabbit hole goes in this case. So not only we see standard neutrophil populations in the lung, but we also see the neutrophil-like phenotype, which actually has healthy dose of eosinophil and precursor eosinophil markers. So clusters that we see here So 0 through 10, they correspond to our bone marrow culture. So we're using it as a template. And this one combines qualities of those three populations. So mature eosinophil, so precursor granule formation checkpoint eosinophil, and the strip1 eosinophil committed population. So absolutely fascinating. It shows that there is plastic population sitting as a resident and yet avoiding detection by all conventional markers essentially, because all markers match except for cyclic alpha and CCR3. So you would not normally even find them if you were looking for conventional markers, and yet we suspect they hold all the eosinophil and neutrophil potential. Now, all those other populations are, monocytes, disease, and so on, so they're different. Interestingly, there is always this one consistent population with consistent set of markers, which is we call proliferative precursors. So MK67 high, CD34, and so on. And there's always consistent detection of eosinophil progenitors. So L5 receptor, GATA2, and a couple of other markers. So now what happens during inflammation? So this is standard of a model. So we all use it. It's 2 sensitizations to chicken egg albumin in presence of alum adjuvant, so 2 IP injections. So once sensitized, you give mice intratracheal, intranasal administration of the same antigen, OVA, and you repeat it, so every other day to promote the inflammatory response. And we know that this model will really mount really robust, eosinophil response. Now, what do we see by single cell RNA-seq? So interestingly, we now see a split in this population. So what I was previously showing as this kind of confused phenotype that had mix of both neutrophil and eosinophil markers, suddenly clearly decides to break apart into strictly eosinophilic. So it has all just markers of EOS and pre-EOS and the neutrophilic. And aside, there is also additional neutrophil population. So then question is, what exactly is happening, right? So are the cells recruited and they're still developing in lung tissue, or do some of them actually develop from those resident populations? Now that's a question of plasticity. So we need to do much more careful work to actually delineate where those convergent phenotypes are coming from and what they functionally mean. So functionally what they mean, actually, I can tell you right away that they express IL-4 very highly. So these cells, which we consider classical eosinophils, and ALOX15 actually. So something that in the first talk we discussed as this resident phenotype, which is pro-resolving, which is interesting because by all parameters we look at this and think this is just our inflammatory eosinophil, right? And it's clearly a newcomer because it was not there in the resident tissue. So another point, just if you remember discussion from bone marrow cultures that phenotypes are transient, just the fact that we can see these markers means that it probably just started actively expressing them. So it's definitely some new event and possibly cells which are being generated as we're detecting them. So another thing we did in regards to single cell, then we said, okay, if we look in public repositories of single cell data, can we actually find eosinophils where eosinophils are not reported? So now understanding that mature markers may be escaping us, can we use some of those alternative markers we came up from those populations? So we picked those 3 here, and this is not something we classically talk about in regards to eosinophil biology. So it's solute carrier, for example, so SLC 7A11, GM5 4A3, and so on. And actually with high fidelity, they map to both, interestingly, eosinophil and neutrophil granulocyte populations. So this is mouse cell atlas, if you know, that's a repository where they put out carefully characterized populations, and they actually classify them as eosinophils, neutrophils. And interestingly, using completely alternative markers, we absolutely come to the same populations. And then tabula muris, they have only one unidentified population, which they call leukocyte, very specific. if they map actually right exactly there. And that was probably the only population missing there at that time. So they did not report eosinophils, but all other cell types. So aside from just this phenotypic detection and evidence that there potentially a lot of plasticity, myeloid plasticity happening in resident state and during active inflammation, question is what actually do they do? What do those different subsets contribute in different tissues? So for me, big interest is their interaction with the tissue environment, right? Specifically extracellular matrix, I discussed that a lot in my talks, how it can activate differential eosinophils and how they can actually either break it down or build it to assist remodeling of tissue. And that's my true interest is and belief that these cells differentially contribute to those tissue remodeling processes. So this, for example, we just fished out those cells with eosinophil phenotypes and aligned top 100 markers from different situations. We took our single cell signature of allergic lung, EOS, single cell of naive lung, and naive bone marrow. And you can see that are pretty darn different in all those situations. So there's very little overlap. So you can see even if we're talking about same phenotype and same population by single cell, like functionally they would probably express different stuff. So that's a work in progress just to sort them out, trace them, describe them. But again, in allergic situation, interestingly, they would express pro-resolving signature, right? And then in homeostatic conditions, it's entirely something else. But also talking about extracellular matrix, you can see expression of MMPs, for example, and serine proteases acting on plasminogen system and so on. So you can see it's a deep involvement in tissue maintenance processes. So to sum it all up, so I keep believing that the eosinophils are tissue dwellers and play this homeostatic and morphogenetic roles, which just come up in different flavors and different remodeling situations, be that injury or some normal developmental event. And unexpectedly, a single cell is revealing just what is the extent of plasticity of those myeloid cells in tissues, the potential they can jump from lineage to lineage. And I think it's absolutely fascinating. And it just opens just so many doors and questions in regards to neutrophil versus eosinophil responses in different situations, for example, and what may be governing that. An implication, technical implication of this analysis that, now we can see from the session, not just from my talk, it's definitely possible we can detect eosinophils, but then possibly only in certain situations based on conventional markers. In other situations, we have to be more creative and think outside the box, because I realized that a lot of those markers may be just silent in resident states and just appear during differentiation of cells or activation or some transient responses. So, and I always say in my talks, and I'll finish this one with this as well, that to fully understand the cells, we just have to look not just at the eosinophil itself, but the environment that's residing in or being recruited to. So those two together can really put things in the context. And we've seen it from those previous, talks, beautifully laid out how different subsets of eosinophils and particular responses played beneficial roles, right, for example. So with that, I would like to finish and thank multiple people that were involved in those projects. So many people rotating through the lab, contributing one or another piece to studies with eosinophil biology. So very hardworking, dedicated group of people. My collaborators and colleagues at Northwestern University, especially Hematopoiesis Valencia, who was instrumental in working out single cell approaches, and Bruce Buckner, always willing to discuss biology of eosinophil, like extremely helpful in all regards. And Bob Schleimer and Nav Chandal, that also discussed aspects of alternative activation of those cells, and others. And last but not the least, Bas Jacobson, who were always willing to provide tools for eosinophil research. And now deceased Jamie Lee, who actually got me started on this road of homeostatic eosinophil biology. And thank you all for your attention. I'm open to questions.
Thank you very much, Sergej. There are a lot of questions on the Q&A forum. So there is a question from Andrew Higham. Great talk, very interesting point regarding the lack of EPX and MBP in mature eosinophils. The question goes, is this full commitment, or can the expression be switched back on during inflammation? Or are these cells, some of them in a senescent state, and might never actually make it to become mature eosinophils?
Yeah, those are all great questions, and I'm afraid we won't be able to answer it even with educated guess without doing tracing analysis of those populations. So more work is needed to actually trace, right? What happens to the subsets that we see? Right. What is the potential, right? Without it, we can only guess.
Right. I have a related question there, Sergej. As you know, the tracing, obviously a good one would be IL-5 receptor alpha Cree lineage tracing, for example. But then, you know, the guess would be that based on your data now that neutrophils might be tagged and maybe even some dendritic cells. So has anybody ever tried to do an IL-5 receptor alpha tracing?
Yeah, it's a good question. Not that I know of. Mm-hmm. So development of IL-5 receptor, any populations of myeloid cells?
Because I guess, you know, the point you were making about that population, even if you do a single cell analysis, you know, if you would get more gene coverage, it would probably still fall apart in more subsets. So
right.
Really seeing, for me, to see a dendritic cell having a lineage trace of IL-5 receptor alpha, then I would really believe it, you know, that it really came through that pathway. That would be for me the definitive proof, you know, that the DCs can come from these MMC populations. There is a related question here also from Ariel Munitz. So it's about you were saying it already in your concluding slide. What is the role of IL-5 in all of this? So are the eosinophils that are dependent of IL-5 different from the ones that are independent of IL-5? And did any of your single cell data provide pointers there?
Yeah, so that's a good question. So I think a lot of this may not necessitate IL-5 at all. So even our experiments ex vivo show that stromal factors alone in complete absence of IL-5 would be sufficient to generate conversion phenotypes and double positive by 6G cyclic F. So I suspect that other cytokines, maybe GM-CSF, coming from stroma, we actually have data showing that they do express it quite strongly, may be able to generate those, right? IL-5 definitely provides the final push, right, to generate what we call its mature activated phenotype. So that's how I see it.
Right.
But then what's interesting, what it does actually for other myeloid lineages, right? So expression on disease, as you asked, neutrophils.
Yeah, B cells. I mean, B cells.
B cells, yeah. So I'm not the only one. I think it's coming up. So other lineages express IL-5 receptor. Now, what does it mean for them? Right. Is it some kind of switchover signal? And yeah, it's interesting.
There was a question also from Ju Q. Thank you for the great presentation. Is there a gender effect on the transcriptome profiles of eosinophils in the in vivo bone marrow single cell data? So, you know, did you see something there like you were showing in your bar graphs already?
Unfortunately, I'm not that rich to do sex stratification of single cell data. Yeah, each sample costs a pretty penny, so exactly there is no luxury to really work out questions like that. But I'm hoping to proceed now with knowing sets of markers that we can chase. I can start sorting specifically those populations, and then I can address that stratification.
Right. And we have a final question from Sjoerd Schetters. Excellent and thorough work. Thanks a lot. Question is like this. The eosinophil turnover is over a week in the lung compared to a day in the circulation. So do you think the eosinophil development niche in the lung is related to the eosinophil niche in the bone marrow? And how would that change during inflammation? So, you know, there's the same question from Steve Ackerman, you know, what happens to those cells when they arrive in the lungs? You know, what is their developmental program? What are the cytokines, and do they need to meet up with a particular partner to make it, you know, as an eosinophil?
That's a great question. So I believe that absolutely yes. they need this microenvironment support, they need partners. And I believe that what we call remodeling and repair provides that platform.
Mm-hmm.
So activity of those systems and the position of specific ECM microenvironments, I think, is the actual clue for those cells to either develop into something or stay in the phenotype that are in. And this is specifically true if you think about gradients, right, of those events in tissue. And that's where you may observe different phenotypes coexisting, because that's what ECM may actually create those micro niches in the tissue. And then cell reaches that point, docks on integrins, and says, I'm going to stay as a remodeling eosinophil, or it's time to finish remodeling and repair, so I'll start degrading ECM, and it comes to what we call a subset, when in fact it could be just a plastic subtype.
So depending on the needs of the tissue, you would adapt more or less.
Exactly.
Okay.
So all
thank you
points
very
to
much.
the direction that it's plasticity of the tissue response.
Right. Thank you very much, Sergej. I'll give the word back to Florence. I think she wants to do the concluding remarks. Thank you very much,
Bart.
Sergej.
Yep.
Thank you. Thank you to the speakers for very stimulating presentations. And thanks to Bart Lambrecht for moderating this very interesting session. Thanks also for attending this webinar. I hope you appreciate it. And in this respect, we will soon be receiving an email with a link to an evaluation form, and we would be very grateful if you could take a minute to fill it in for us. It will help us improve our future webinars that we will plan throughout the year. As you may know, As you may know, the next webinar will be held on Wednesday, April 28th, same time as today. And this one has been programmed in memory of the late Barry Kay, whose contributions to understanding eosinophilic airway diseases are innumerable. Please also note the dates for the next webinars that will systematically be held on the last Wednesday of each month at 9:30 a.m. Eastern Standard Time. And don't forget to submit an abstract before May 17th for our special autumn abstract session. Thank you to everyone. I think it's time for us all to go back to our daily activities, and I wish you all a wonderful day.